Okay, we start. And thank you for being here. And today we have Dr. Paula Martinetto of the University. Paulina, what did I say? Paulina, sorry. I'm tired. De la Universidad Nacional de Mar del Plata. Y she's doing a 3-month internship here, in research state, with the climate change group, in particular with Novia Marva and her group. So thank you for being here and thank you for
Okay, well, thank you everybody for spend this morning here to listen a little bit about the ecology behind the blue carbon in some marshes in Argentina. I will give the talk in English because the organizer asked me to do it in English, but my English is far from perfect. So if you don't understand anything, please let me know and we can figure out the way to understand each other. Well, yes, I'm Paulina Martinetto. I'm from IMIC, which is the Instituto de Investigaciones Marinas y Costeras. It's an institute from CONICET, which is like the CSIC here, and the Universidad Nacional de Mar del Plata, where I did also my PhD and all my career. I work in the Laboratory of Ecology since the last 20 years. And I'm going to present today some of the studies that we've been doing in some marshes in collaboration with Nuria Marva, who is the person that I'm working here in Medea from the Laboratorio de Cambio Global. So for the next 30 minutes, what I'm going to talk about is about blue carbon and the ecological characteristics of the Southwest Atlantic sand marshes. The blue carbon in sand marshes in general, and the blue carbon in this in all sand marshes in South America. And specifically, I'm going to talk about the biotic and abiotic drivers that lead to the blue carbon stocks that we have documented. And I will finish with some final thoughts that we can discuss among everyone. I know that probably there are some people that work in blue carbon, but just to be clear, what we are talking about blue carbon is the carbon that is stored in coastal and marine ecosystems. Specifically, is referred to the carbon stocks in sand marshes, mangroves, and seagrass meadows. And some other ecosystems like kelp forest or intertidal bird areas are also being trying to be incorporated in the blue carbon, but they are still in discussion. So still specifically and strictly is sand marshes, mangroves, and seagrass meadows. And these are important because they, even when they cover less than 2% of the total ocean area, they can account for around 50% of the total carbon sequestered in ocean sediments. And this happened because in these places, the carbon burial rates are very high. This figure shows the carbon burial rate for terrestrial forest, boreal, tropical, boreal, and temperate. And these are blue carbon ecosystems and mangroves and seagrasses. These are burial rates in average. And the scale is logarithmic, and you can see that these ecosystems have an order of magnitude higher burial rates. The carbon that is in the sediment also can stay there for very long terms. These are two examples. This is an arrecife, a reef of carbon in Formentera near here, that was exposed by erosion. This is seagrass meadow. This is Posidonia, and all this reef have been stored carbon for those 10 years. And this is another example in the Bay of Fundy in Canada, in Salt Marsh. It was also exposed by erosion for this tidal channel, and this contains carbon that lasts for 3,000 years. So it converts these systems in important areas. And under the climate change scenario, these ecosystems became very important to face climate change. this is not something that we didn't know before, but it's just that we call it blue carbon since the climate change scenario. So what are the keys for this? This happened specifically because, or mainly because this ecosystem have high photosynthetic rates, and also because they have a large production of roots or underground biomasses, and this can facilitate the capture of particles and suspended sediment that come in with the tides. With every tide, these are intertidal areas or submerged areas in the case of the seagrass meadows. These water columns have a lot of sediment and particles, and they can precipitate and be trapped in this root system. And what still amazes me is that they are very different in terms of structure, no? Because this is a forest, a real forest. This is a meadow, is Marshall meadow, and this resemble like a grassland, no? You can see it are much like a grassland, and they still, the three different systems, they can store a lot of carbon, and they develop this intricate architecture of the roots. And the other key is the low decomposition rates that happen here. These soils are mainly anoxic, so the decomposition rates are very, very low. And in addition, sometimes they have sulfur contents, so the root for the methanogenic bacteria are somehow inhibited. So there is no release of methane, which is another of the greenhouse gases that worry everyone. And all these make that the system can preserve the carbon for in a millennial time scale. So the salt marshes that we have in Argentina, well, they span the Southwest Atlantic. I want to refer specifically to the Southwest Atlantic salt marshes, which is the 98% of this, where the 98% of the salt marshes in South America are. And they span from for those 300 kilometers of coastline from Rio Mampituba in the south of Brazil to Rio Grande in the southern Patagonia area. So this is the area estimated, almost 220,000 hectares, and the 95% is located in Argentina. And they are dominated specifically for three species: Spartina densiflora, Spartina alterniflora, and some species of Salicornia that right now is believed that we have two species, but still is in doubt. So I will refer as Salicornia. And the other main characteristic of, like a feature characteristic that I will present today is that they are densely inhabited by a burrowing crab called Neoelis granulata, which is an herbivore and a bioturbate. This crab inhabits salt marshes and the mudflat in the intertidal area. This is a picture of the mudflat with low tide, and each of these holes is a burrow. So you can have an idea of the bioturbation that this crab can have in these systems. And in these systems, this crab intervenes in several interactions. This scheme summarize some of the interactions that we have been established in the group. As you can see, there are trophic competition and non-trophic positive and negative interactions with many different components of the ecosystems. And I will refer today only on the salt marsh part. In the salt marsh, they inhabit in the salt marshes that are northern than 42 degrees, which are 98% of the Southwest Atlantic salt marshes. This is around Peninsula Valdez in Patagonia, in northern Patagonia, Argentina. And in these places, they can have densities of up to, it's over 100 burrow per meter square. We have counted like 120 burrows per meter square in some places. This influences most of the ecological processes in the sand marsh through herbivore and also by bioturbation. So as a bioturbation, it can remove up to 2.4 kilograms of sediment per day. So this reworking of the sediment redistribute all the detritus in the sediment column. And also the burrows can reach up to 1 meter. So they, remember that I told about the anoxic soils in this system? Well, there is no anoxic anymore with crabs. They inject a lot of oxygen with the burrows. So this increase aerobic decomposition and also change the root shapes. So the architecture of the root of the plants change from an anoxia tolerant to a nutrient efficient uptaker. These places are characterized by being limited in nitrogen, so being an efficient nutrient uptaker is a good thing for the plants. And because these soils have oxygen for the crab, the mycorrhizal associations with roots can be possible, so they can be efficient nitrogen uptakers with this association. So for you to have an idea what is the soil, is like a cheese, it's like an emmental cheese. I usually say in Argentina a Gruyere cheese, because in Argentina the Gruyere have holes, but I learned here that no, the Gruyere cheese doesn't have holes. So this is like an emmental cheese. So also as a bioturbator, the burrows can work like real traps of sediment or organic matter and all the particles that are suspended in the water column. The burrows have the entrance of the burrows have the shape of a funnel. So all the particles that are in the sediment can precipitate in the burrows. But also the detritus also that fall from the plants, but also contribute to the erosion, because as you can see in this picture, all the holes that are in the sediment make some vulnerable to the force of the tides. So it has been documented that the burrows contribute to the formation of tidal channels in the sand marshes. And as an herbivore, it consumes large amounts of plant biomass also. I mean, they are very abundant, and when they feed on plants, you can also even listen the crabs within the plants when you work on the sand marshes. And we have documented reduction of up to 40% of the biomass. But in addition, the leaves that are damaged by the crabs when they are eating, they can easily be infected by fungus, so this increase the senescence rates. So back to blue carbon. What we know about the blue carbon in sand marshes at global scale? Well, this is a map from 2014, so it has already 10 years, showing in gray are the places where sand marshes have been documented, and the colored one is the places where we have actually data of blue carbon. And even if the data since 2009, where the blue carbon concept was released, has been increased, the data is always biased to the northern hemisphere. And even if we update this map today, you can we can have all colored here in Australia, but still all these points are in gray. And another characteristic is the large variability that we can find in places. So these are average of carbon stocks and CBR, which is carbon burial rates. And the average published in 2022 were 370 tons of carbon per hectare. But if you see the range is between 27 and 1,700. And the same for carbon burial rate, which is in grams of carbon per meter square per year. the average is 168, but the range is 1.2 and 1100. So we know that there is even if they all these places are carbon sink areas, there is a lot of variability, and that is handled by the average, no? And the mechanisms that are behind this variability has been rarely explored. I mean, since the release of the blue carbon concept, and because the blue carbon was think in climate change context and aimed to manage areas with that are important for like carbon sink. Most of the studies just document the values of stocks and burial rates, but rarely go behind the mechanisms that are driving these values. And in general, when they do, they focus on environmental variables like granulometry or temperature or kind of more general variables. However, the biological variables has been very addressed, even when there are many studies that document that they can be very important for carbon cycles at local or at patch scale. No? So if we think in the carbon transformation pathway in a given sand marsh, we can summarize the pathway in this scheme, where you have the leaf biomass by plants that uptake carbon by photosynthesis and convert this carbon in biomass. No? This biomass can be consumed by consumers, and part of this carbon is back again by respiration. Some other part of the leaf biomass became senescent. So it confirmed this dead biomass above ground and below ground, and this entered in the decomposition routes or path that can release eventually more CO2. Part of this can be also exported to other systems, and part of this is finally buried in the soil and became blue carbon. Some the adjacent systems also contribute with some organic matter. But when we add crabs in this scheme, we see that it intervenes in all the links that we have in the scheme. So the crabs, by herbivory, they consume leaf biomass, so they reduce the biomass that is ready for uptake CO2, and also increase senescence by the facilitation of fungal infection. Through oxygenation, is increase the decomposition, because bacteria that are aerobic can have place here now, because there is oxygen. Also, the oxygenation of the burrow, as I say, allows the association with arbuscular critical fungus, which increase the leaf biomass. And by bioturbation, they can increase or decrease the burial. They can modify also the exportation and the importation to other systems. So, what we know studying this system for many years is that evidently the presence of crab will have a large impact on blue carbon in these systems. So, given the documented worldwide variability and ecological effects that we know about crabs, we hypothesize that these crabs could have a large impact on the blue carbon of the Southwest Atlantic sand marshes. So to address this, we set these three objectives. We look to determine the carbon stocks in soil and in biomass, and the burial rates in the sand marshes in Argentina. Also, we look to evaluate the role of biotic and abiotic variables in the blue carbon, and we update the global estimation of carbon stock and burial rates. So we conduct a large field campaign from the Buenos Aires province northern to the Buenos Aires province in Bahía San Borondón to Puerto Loyola in the Santa Cruz province at the end of the Patagonia, Argentina. We select 11 marshes, which were the most representative of the region. And we strictly study the ones that are covered by one of the three species, the dominant species. So in total, this span for 3,000 kilometers of coastline between 35 to 51. So we have a nice gradient of environmental variables as well. So I summarize the methodology in this slide. I mean, it was a lot, but I try to summarize in here. So we, in each site, we first identified if there were two or three of the dominant species, and each of these areas were 5 replicators. And in which replicate, we take associated data of above ground biomass that we separate in green and senescence, and we measure carbon and nitrogen contents and the isotopes of nitrogen and carbon to look for the contribution of the plant itself to the blue carbon. We also measure burrows, we counted and we measure the diameter, and we calculate the volume to correct the carbon stocks for the empty volume occupied by burrows, because if you can imagine these emmental sheets with a lot of, there is a lot of empty space. And when you do not correct for the empty space of the burrow, you are overestimating the amount of carbon. And then we sample also superficial sediments for carbon and nitrogen isotopes to look for the allochthonous and autochthonous sources, and the below ground biomass, again, as the above ground biomass. And finally, we take a core of up to 1 meter depth, depending on the heaviness of the and how far the soil goes in the sand marsh. And measure organic content. We slice the core from 0 to 5, 5 to 10, and then every 10 centimeters. We also calculate the burial rates, taking cores in the upper and the lower areas of in each site, dating with plumb 210 to measure the sediment accretion rates. And also we analyze granulometry in this course. And with all this information, we run a path analysis, SM, a structural equation model that includes biotic and abiotic variables. So we include the data that we took in situ, plant species, crabs, burrows, plant biomasses, carbon and nitrogen ratio, granulometry, sediment accretion rates, and the carbon stocks. And we collect data from databases, precipitation, temperature, and tidal range. The tidal range in all these posts that we sampled range from 1 meter to 13. So we have an amazing range of tidal influence. And it's semi-diurnal, so we have the enter of this mass of water twice a day. So the results. The results was, of course, variable. These are the stocks of carbon in each compartment. In green is the above ground, green and senescent, and in blue is roots in dark blue, and the carbon in sediment in light blue. And these are each of the sites. So we find a range between 6.47 and 111. And we also found that the green above ground biomass can explain 10% of these carbon stocks, and the senescent is less than 2%. But the roots, you can see in these colors here, can represent up to 40%, specifically in Spartina alterniflora and Salicornia salt marshes. With the isotopic analysis, we found that a high alloctonous contribution, which was very important to note, specifically in these salt marshes in the Buenos Aires province, where POM contribute with between 80 and 93% of the stocks of carbon in these salt marshes. And in all other salt marshes, all the contributors were equal. So there was no one dominant contribution. So it's important to know that the alloctonous material that arrived to the site is a big part, a large part of the stocks of carbon in these salt marshes. So in average, the carbon stocks for all these places was the 42 tons of carbon per hectare. And in total, considering the area occupied by the salt marshes, is about 4 million tons of carbon sequestered in these soils. And most of this carbon is below ground in sediment and roots. Regarding the burial rates, they were also variable between 7.38 to 204 grams of organic carbon per meter square per year. And in total, considering all the area occupied by salt marshes, these salt marshes are capturing 55,000 tons of organic carbon per year. So for the same analysis, the good news that we have is that we found a good fit. It is a very good news when you run this kind of analysis, because it's not something very easy to find when you have a variable so variable. And also the good thing was it revealed that a combination of biotic and abiotic variables explain a large part of the blue carbon, the below ground organic stocks. And the variables were the identity of the plant species, the granulometry, the sediment accretion rates, and of course, the grass. So with all these results, and we also compilate the information from the literature to update the global average of blue carbon. And we recalculate using 743 points that the carbon stocks in salt marshes soil around the globe now is 185 megagrams of organic carbon per hectare, which is 41% lower than previous estimation, which is a lot lower. But when we start to compilate all this data, we found that most of the data that are very documented in the recent years are lower than the first records. So it seems that the first records just document the most large ones, and now with the incorporation of more data, it seems that there are places with less carbon, organic carbon. It still is a lot of carbon, but it's not like 300 like we have documented before. And the same, some of less, the same happened with the carbon burial rates. We re-estimate this carbon burial rate in almost 200 grams of organic carbon per meter square per year. And this is 6% lower, which is not a lot, like 41 in the carbon stocks. And well, the range is, the lower range in carbon stocks and in burial rate was in Caleta Los Loros in Argentina, so we have the lowest record. And the largest are in New Jersey and Louisiana salt marshes. So in conclusions, well, first of all, the importance of the biotic variables in these systems. the ecological studies have provided many examples of the importance at local and patch scale. But it seems that they were only studied at that scale and never been incorporated in regional studies to explain the variability. It seems that looks like something impossible to do it. However, we incorporate the biological variables in this regional study, and we found that they can be very important driving the blue carbon. The second is the importance of the allotment sources, and these have a very big implication for restoration, considering the connection between systems, because if between 80 and 93% of the carbon in some of the sand marshes that we studied is coming from outside, the management of this area needs to incorporate this, because otherwise it's no will be not successful in management, or only considering what happened inside the sand marshes. Which is also very interesting is to study the mechanism of autochthonous carbon losses. So what happened with that carbon, because the sand marshes are very there are ecosystems that are very productive, so they are producing a lot of carbon. So part is consumed by everybody. Some other part can be exported. But it would be interesting to analyze what happened with all this carbon, where is going this carbon. And we are running some experiment now with the student that is study the interaction between photodegradation and the effect of tides. And we are having a very good results showing that photodegradation is the main driver, also the loss of carbon in the atmosphere. And the third and final point is the overestimation may occur when you don't have specific data from the site, no? For instance, we use the 370 average to estimate the carbon that we have in our sand marshes. It will be overestimate. So we need to be very careful with those estimates, specifically because we are considering the role of this system to face climate change. So we need to be more careful with this data, with the use of this data. The same with the equations that convert organic matter to carbon. In each of these sites, we found different relationship between organic matter and carbon. And in some places, in some studies, they or even in the blue carbon manual, they recommend if you can not measure carbon specifically, you can convert from organic matter. And we found that it's not so easy or so accurate to use that. And third, the other source of overestimation in our case, and in many cases where bioturbation is a big issue, because it's not just in Argentina that we can find this. All the coast along China, for instance, is covered by crabs, and bioturbation is something that they are studying that is the main driver of blue carbon too. So the lack of correction for empty space by burrows can also lead to an overestimation. And we found in some cases that it can be between 20 and 30% overestimated if you do not correct for the empty space occupied by burrows in some of the sand marshes. So some final thoughts. These are many of my students and my dear colleagues that work with me, and we are a nice group. I mean, we work very well together, and for me, I've been here for over 20 years working, and I'm happy to work with all these people, which is a very good thing, no? I'm very fortunate for that. And we do ecology of sand marshes since many years. And we know that at the end, all the interactions that happen and all the ecological functioning is driving the blue carbon. we knew it that before to read about blue carbon. So my final is always don't forget the ecology behind these systems. And the case of ecology studies in this ecosystem lay the groundwork. So it's the background for the development of the blue carbon concept. The blue carbon concept doesn't could exist, it doesn't work for the ecologists driving ecology many decades ago. And because we are dealing with natural systems, I think that ecological knowledge is where the key lies to understand the variability we find in nature. In terms also of blue carbon and management, understanding the local ecological processes that govern blue carbon gains and losses, because the studies always focus on the gains, because we want to store a lot of carbon, but we need to understand also how this carbon is released to other places, to the atmosphere, or gone to adjacent systems. So the what govern losses is as important as the path. And this is important to predicting ecosystem response to global change and implementing successful climate nature-based solutions. And with that, yeah, I have to say thank you to UIB, because I'm here because they will give me a fellowship. So that is the reason, because I'm here. I'm very, very happy to share with the people here and to learn about the Mediterranean ecosystems. And for Fortune, science is collective. And I need to thank all these people that have been working with me for many years. And Ecology, and Eric Spark from the US, Nuria Marra, the main reason because I'm here today, and Diana Montemayor from Argentina, Raymond Ward from the UK, Just Cebrian, and immigrated to the US, Juan Alberti, and Oscar Ibarne, and this is a wonderful group of people that I need to thank. And Montes Casis, and these are the founders so far that we have in Argentina. I'm sure you have listened some bad news about Argentina. We are in a very dangerous and very sad situation in terms of science and university and education, but also in social. So these are the three sons of fungi that we used to have, the CONICET and the Universidad, and this was the Ministerio de Ciencia that is already gone. So I don't know what result can I present in future years. Hopefully we can be still working as these years. Thank you.
Tengo una curiosidad sobre cómo hacéis exactamente para saber cómo son
Las pruebas.
las cuevecitas de los cangrejos. ¿No sé si pones pasta o algo?
Bueno, hemos hecho con pasta para ver, continuing. I don't know if everybody speaks Spanish or there are anyone that doesn't understand. Okay, so I will continue in.
And you can
Yes.
translate it.
She asked me about how I measure and look for the shape of the burrows. So what we do is in former study, with some I don't remember the name in English, but it's like a foam that gets solid at some point. So we found that in the sand marshes, the burrows are striped, so it's very easy to and they go more or less equally the diameter to the bottom, and they go until the end of the soil. So if you sample the whole soil of that you have in a sand marsh, you are covering all the length of the burrow. In the intertidal bare area is more difficult because the shapes are different. So is either, I mean, it's not so strive for the conversion or to do a correction for volume. But in the salt marshes was quite easy.
Thank you, Paulina. It was very interesting because it covers so many
I forgot
topics.
the
Yes. The extension of the study, the lack of information before, no? And I'm curious about the crabs because we've been always in the carbon cycle thinking
Yeah.
that the below, well, the above grass, no, was
Yeah.
impacted by herbivores,
Yeah,
but it's very low compared to other interactions. You know?
Yeah.
So perhaps impact in below.
below and above.
So
Yes.
it's very interesting. And also I remember in Florida, we have crabs too,
Yeah.
impacting the grass.
Yes.
So it's quite interesting. But
Yeah.
do you have any sun marshes not impacted by crabs?
Yes, the southern ones, because
Okay.
crabs distribute up to northern Patagonia.
Okay.
So the sand marshes were, I mean, we also have a gradient of crabs.
Okay, so it's
No.
from more impacted to less
Yes,
impacted?
yes,
So
yes.
you can also compare different places, the
Yes.
carbon stock in one place to
Exactly.
another.
Yes,
That's interesting.
and even we are trying to, it's very difficult to do exclusions in
Yeah.
film, because crabs are very
They
smart.
can destroy,
Yes. Yes.
I've been working with that and they destroy the whole cage.
Exactly, yeah,
They
yeah.
eat it and they make holes and.
But we have some areas we manage like low and high abundances.
Mm.
So just taking crabs and keeping some low
Okay.
density in some places. And this also, the former studies that when I showed the network, the carbon transformation pathway affected by crab, these are all different studies, local studies that we were doing to develop the hypothesis, the general hypothesis, no? And in those cases, we exclude or manage the
Density.
yes, yeah.
So is it a local, is it an endemic species or?
Yeah. No, it's endemic. It's, I mean, it's native, but it's not endemic. It's not like just happening here.
Okay,
It distributes, or, well, not sure if the China one is exactly the same species, but it's the general, same general. Yeah, it's a graph.
and it happens something for the increasing population of the crab? Why? This is
Why they
happening?
increase the population? We have record some increases or decreases related with El Niño,
Okay.
because El Niño is a big driver in Argentina. El Niño is very wet, and the La Niña is very dry. So when we have extreme Niños and La Niña, we have these conditions very marked. So we have recorded some variations in the populations coupled with the Niño and La Niña events.
Thank you.
Thank you so much for it. It's been extremely interesting.
Thank you.
So it's just a question because about the environmental parameters that you include. We are starting, as you know,
Yeah.
working on some marges. And we discover in the preliminary data that the salinity
Yes.
is a factor that highly controls the.
Yeah, we include in this SEM, we included like the contribution of fresh water.
Okay,
Because some of the sand marshes are more estuarian, so they have a large contribution of fresh water, and the other ones are more in bay areas or riparian areas where we know like a big contribution from fresh water. And this, I mean, this is like a proxy of salinity also. The Patagonian one that are more, they have also less contribution of water, they are more salty. So it was also a way to include the salinity. And the other thing that was I mean, include somehow the salinity is the plant identity, because the three species are very selective and have different competitive abilities with different salinities. So when we decide to include the species, the plant species, was not just to include the name, but because the establishment of one or the other include a lot of variables itself. No, so it's not just a name, but an ecosystem.
It has correlation with
Exactly.
environment.
So yes, and so to arrive to that same, we tried a lot before, no? And we include salinities and we include some other variables first, and then that was the model that we decided to, because there was a lot of correlations that made noise in the interpretation.
So my second question is more asking us an advice.
Okay.
Is that we have some problems with, we are comparing also species because in a gradient of salinity, just having this correlation. So we have problem with some species taking the course because they are in a sediment that is really, really dry.
Yes.
And the course are always broken.
Oh, but like what kind of
with
current?
fungus?
Yeah, with fungus, yes. Yeah, we have fungus in more fresh water areas, no? But I
Which
use
material did you use?
we use it because some of these marshes have very bad. We use a Russian core, a metal Russian core, and
Yeah.
in some places, yes, yes. I think that it would be the best.
But it has no problem then with data when you want to do the
For compactation,
when you took the core for
Yeah.
data.
Datation with
With the station.
no, but for dating,
Better.
for dating, no,
Yeah,
with
that's
the
the point.
plastic, PVC ones.
Okay,
No, that's for carbon stocks.
okay.
The other way, yeah, no, for the others, you have to, but because you don't need to go. For dating, we only need like 20 centimeters, because then the signal of the plum is gone. So 20 centimeters was enough for us, so we put all the effort to to take this, taking care of the 20 centimeters.
Thank you.
On the global scale analysis, I'm not sure I understood, if I remember well what you said. You have lower levels found compared to previous studies,
Yes,
and you said it's most likely because in the previous studies, they only use the,
I think there is a bias, yes, it's a bias in the report of, to the time. There is a bias in the report of the studies with larger stocks at the beginning.
because the area of study have larger stocks and while in the studies nowadays, they use more areas.
Not for the area itself, it's because this is, I mean, when you report, you are reporting the data in organic carbon tons per hectare, so it's corrected for
Yeah.
the area.
I mean,
it's-
the study sites. So
Yeah, for the study size, yeah. So I mean, I have not checked this, but it's, along the time, when this global estimation have been updated every 2-3 years, 5 years. And if when you see the history of this reporting, you can see that at the beginning were like extremely high, and then starting when more data is incorporated,
Because the variability.
the variability increase, and the variability increase.
Yes,
To
because
the,
you're including.
down,
the
not
other
to up,
yeah.
to the upper.
Not always
The distribution.
nice
The
and
lower
lower,
part of
nice
the
and much,
distribution.
but it can be impacted or not. No?
Yeah.
That's right.
but then it's not comparable then, but could you compare only the sites that were used 10 years ago and the sites today, only these, not using more data on different study sites, just to see if the bias is related to the introduction of more sites?
I didn't analyze specifically. I mean, that is an hypothesis that I mean, looking at the data reported along the time, you can see that I didn't study to corroborate that hypothesis, but it seems possible at least. Sometimes happen with these new things, no? That
It's about them.
no. I mean, I have to be clear also that sand marshes are good sinkers or carbon sinkers. I mean, still they sequester a lot, but we don't need to overestimate. They are good carbon sinkers. So let's admit the variability that we can have in the data, because that teaches us a lot on the mechanisms behind. Any other?
I had other questions, but it's already almost one. Maybe the last one, on what you said on the photo degradation with the tidal, intertidal area.
Yeah, because, well,
What is it exactly?
this is actually, well, it's actually one a picture. This is the experiment. And we set upper beds and low beds, simulating upper and lower sand marshes. And each of these liter bags have Spartan liter, and a filter of UV blue-green, or no filter. And we measure how much carbon is released in the upper and in the lower, and we found that the combination of because photodegradation is the primer for degradation. I mean, plants need photodegradation to break the cell wall in the plants, so the bacteria and the fungus can enter. Otherwise, they are not able to enter and degrade the tractus. So we use photodegradation, but when it's the effect of the photodegradation happens, the effect is huge. It can wash the carbon from the cells and taken out of the system. And that's not happen in the upper marsh. So it's also a source of variation, because if you sample in the upper marsh and in the lower marsh, you can find different things. And considering the range of diets that we have in Argentina, this is something that can be very important. I know here you have diets, so this is an issue.
Very interesting.
Okay.